Yeah, it's little bit different from whatever we are listening up to now. I will talk more about cell protein synthesis and decision making of a single cell. Then we'll move to collective decision making of a multicellular setup. And this is one of my work with my doctoral supervisor, Dr. Shayanturi Ghosh. So as a whole, I will talk about mostly focused on microRNA, starting from very simplified version of how microRNA regulates gene expression, then moving towards one of the feedback regulation, how it changes that dynamics, then towards sharing of microRNA, how as a whole the things change from single cell to collective multicellular setup. So let me just explain the audience is diverse. So what microRNA is. Before that, let me explain what gene expression. So that is basically protein synthesis. And we know that protein do almost all the activity of a living cell, from metabolism to stress response to communication. So people are really fascinated to understand how and when the cell know or which protein need to synthesize. So there are different regulation in gene expression. So if we again consider this pathway, it can be just considered largely in two steps. One is transcription, one is translation. Transcription is something like my DNA information is copied to messenger RNA, and then in translation, the protein is synthesized. MicroRNA is one of the cellular ingredient that comes after transcription. It binds to the mRNA, it makes a complex and it degrades the mRNA. So in simpler words, where is the, oh sorry. So if I want to mention that how the things are happening, just in very simplified version, say I have two microRNA in my system and one mRNA. So with all equivalent thing, I consider one microRNA binds to one mRNA, it will be degraded, there will be no protein. Similarly, maybe when they are both in equal number, there might be no protein. Once the number of mRNA is greater than microRNA, there will be some mRNA left which eventually translated to protein. Almost in a simplified version, it looks like this, that say I have 100 number of microRNA in my system and I'm increasing the transcription rate of my mRNA. So up to I have 100 mRNA in my system, there will be no protein, so it almost zero. There is a kink and eventually it gives a linear response. It is called the threshold linear response. And why it's important, the cell, it helps cell to remove fluctuations and remove short information to just there will be something like removing those things. In case of 2D array, the same thing is done. Like I have my arranged 40,000 cell here, so I am increasing the target transcription rate in this graded response. The maximum protein will be synthesized here, and up to the mid or something, there will be no protein synthesized when there is mRNA rate transcription is lower than that of my microRNA. So moving to the feedback regulation. So let me just tell you that feedback regulation is very common in case of gene expression system, where the synthesized protein sometimes regulates positively or negatively its own product or something like that. So we considered one non-cooperative positive regulation, that the synthesized protein, if you see the mathematical form, that it increases with increase in U. So it activates or positive regulation to the synthesis of mRNA. And one thing to mention is that there is no cooperativity. So what do I mean by cooperativity? That sometimes they forms dimer and then that regulates further synthesis. So there will be one N in U to the power N by K plus U to the power N. If N is 1, I am saying it non-cooperative, which I considered here. And in case of non-cooperative system, the system is not enough non-linear to give me bistability or something like that. So with this, I proceed that there is some binding of microRNA with my mRNA. There will be some unbinding rate as well, and there is a complex formation which I considered. And to remove the other complexity of the system, I consider whatever my mRNA that linearly translated to protein. Now, there is lot of parameter. We reduce that set. We came with a single equation which actually contains all this information. To analyze it initially, it almost looked like a quadratic equation. We reduce those things, get the discriminant, and find that for a certain region, we find that discriminant is positive. What does that mean? There is a region of bistability. So initially, my microRNA regulation only gives me threshold linear response. Once I considered the non-cooperative positive regulation, which alone cannot give me enough nonlinearity is the system, together as a whole make the system bistable with respect to a parameter alpha, which almost we can consider that how much the positive feedback it's giving to this protein synthesis. The bistability curve almost look like this. With respect to different parameter, we can again say that how much the switch robustness is. This is more kind of the magnetic system and the hysteresis we see in those systems, that it maintains a positive and forward and backward memory. Those kind of things are all applicable here. So moving to the more fascinated part of it, where I simulated the system for a 2D 40,000 cell. I initiated it with a random initial condition and allowed the protein to diffuse. So what I see with respect to time, that the two steady states the cell achieves, and then there is a transient patchy pattern formation in output. So it's not the end state. So we can argue that it's not the end state of your system. It's eventually going to this low synthesis state. Then why you are interested in it? Because the time of this transient, and we are considering a biological system where the genes are connected to the downstream. And in case of development, we are not only interested in whatever the last state is, rather than how the system reaches the end state. So in between time, how these genes are regulating the downstream, that might be very interesting to observe when we connect it with those things. As well as there is a phenotypic heterogeneity in the population, which is completely driven by the underlying bistability of the system. These patterns are not those kind of Turing pattern. There is no Turing instability or something like that. The underlying bistability with diffusion for a particular parameter design is giving this kind of pattern in the output. So another thing, we cannot just ignore the possibility of different positional information in our system. So I explored it in different positional information, how the things are changed. So it's a sinusoidal initial condition. And I can see that with respect to time, the pattern gets we getting a stationary pattern in output. So that is again establishing something, that regulation of microRNA, which again getting a feedback, can give a complete different output when there is no feedback in the system. So I considered it with respect to some randomized, like not the particular value, it is like the sign. Oh, do I have it? So I call almost this as stochastic condition. Yeah. Okay. So next, moving to the sharing part, which I want to focus more. So just let me explain it from. So initially, what I say I have 10 number of microRNA, and if my number of mRNA is 7, there will be no protein synthesis because 7 mRNA will be just blocked by microRNA. So this is my pool A. Now I say I have another pool B of mRNA, which also has 7 mRNA. Now, if they individually regulate A or individually regulate B, there will be no end state protein. But if it regulates both, say I have then divided as 5 and 5, so 2 of mRNA will be left, which will finally synthesize some protein. What I have considered that my first mRNA pool that is giving the bistability previously, this microRNA are now regulating another mRNA pool, and if you remember the previous equation, this non-cooperative regulation as a whole giving the bistability with the non-linearity I have with the microRNA regulation. But in the second pool, I only considered the microRNA regulation and no more regulation of non-cooperativity or something like that. So this pool, I expect to give a threshold linear response, but the bistability of the first pool is reflected in the dynamics of the second, which is completely shared via the sharing of the microRNA. So why this is important? This is actually giving us some intuition that we generally consider the modulation of the synthetic circuit we are actually considering in the experiment. So in case of synthetic biology as well, we consider whatever the circuit we are focusing, and that is particularly isolated from the rest of the environment. That is not true. That is collecting for its own synthesis different resources from its host, and only this sharing brings the dynamics completely different than whatever we expect when there is nothing else to do with the different regulation term. This is entirely regulated via the sharing of the resource, and how much strong the sharing is. To focus on that, I pre-patterned the initial pool like this. This pool I pre-patterned with exponential condition, and I can see that the second mRNA pool that is showing pre-patterning or something, this pattern is followed in the second pool of the synthesis of the mRNA2 protein. So the sharing is enough strong that not only regulates the bistability or bistability based pattern, the initial pre-patterning is also reflected in the system. So this is almost the end. So what I try to establish is that the different resource that we are sharing inside the cell that actually connects different individuality of gene expression that takes the decision not only for a single cell but also in a collective cellular arrangement. Individual low state or high state is getting affected to make a pattern in the output. This kind of pattern is also, I am very fascinated to get the mathematical background. We are doing something with the traveling wave analysis and other things, but the effect of this initial pre-patterning and other things are also left to explore. And I would like to mention that the initial transient pattern what I have shown, that also we are getting in some systems, especially in case of cancer system, that is going to stable, that is giving us some stable pattern in the output, which is entirely driven by when the resource sharing is present and not when the resource sharing is not there. So in case of cancer system, when we are studying those things, resource sharing, we know that hypoxia and other conditions, there is very important to understand how the dynamics change. And I believe this kind of study will help to explore more in those kind of systems. So these are almost I want to say. These are some references. This is our team. Thank you.
I have a question regarding what you said as decision of the cells. The way I understand this in biology literature, is that when stem cells differentiate into various different types of specialized cells like neural cells or other kinds of cells, right? Here, that is not the kind of cell decision that you are talking about,
No,
or can
no.
this be applied, extended in some way that you hope to apply it to that kind of cell decisions?
Okay, so not the perfect decision, whatever stem cell decision making you are saying is true. I am more fascinated with the disease scenarios currently. But in case of development, when I have some literature aspect where resource sharing is true, that time also maybe exactly not this, but maybe that I can extend my study to those systems as well.
No, see, the decision is made by that particular cell. I could not see here how the cell is deciding.
So, okay, yeah, you were right, maybe. But what I mean to say is that when there is no resource sharing, there is the monostability, and then when there is resource sharing, there is bistability. So cell decide from one state to one low synthesis to high synthesis, which is it wants to go something in that perspective. Okay, so it's.
I have the microphone. Hello. I have missed something. So in your model, you are supposing that messenger RNA diffuses from one cell to the other. Is this something that is, yeah, I mean, did I understand correctly? Because it doesn't seem...
I consider the protein diffuses, not the messenger RNA. So.
Okay, sorry.
Yeah.
Okay, thank you.
You want to come with the mic or? Yeah. I just wanted to know, when you say you do not see in your first part Turing patterns, is that so because you don't have 2D equation? Is that the only reason, or?
No, we have done. There is no Turing instability in this kind of system.
No.
So
No. But what I'm asking is, you had two-dimensional equation. I forgot the equation. Could you go to the diffusion equation? Yeah. So do you expect? Okay. But why is Turing not coming up? I mean, is there a reason, or is just mathematical?
Turing instability in these systems.
No, that's what I'm asking. Why is that not there? I mean, what should I have changed that Turing would have appeared? Let's say just mathematically.
So this. What should have I changed? Okay.
Okay, maybe I forgot all these things. I just wanted to brush.
So activator inhibitor system, whatever I remember, those are more to Turing system, but I cannot directly answer it now. What should I change? Maybe I will also
because
check.
It has a reaction part, it has diffusion
part.
part.
I understand, but we checked if it has, because whenever we first get the pattern, we consider something like that, but there is no.
If you include cooperativity, it might come up?
No.
Even then, no. Okay. Thank you.
Thank you.